We are going to be talking today about harmful algal blooms with Dr. Asher Schnetzer. Asher came to us in 2012. She's in the Marine Earth and Atmospheric Sciences Department, and she's my advisor. And she has a very broad spectrum of research from both freshwater and saltwater. Her general research areas involve both anthropogenic stressors that have affected microbial ecology, diversity, plankton food web dynamics, and harmful algal blooms. She has conducted research all over the world, ranging from the Mediterranean, the Sargasso Sea, the Southern Ocean, the North Pacific, and including the Deep Ocean. So let's welcome Dr. Schnetzer.
Right, there's a clip I need to push, right?
Yes, excellent, perfect. Hello everybody, thank you so much for the invitation, and thank you Gabrielle for this wonderful introduction. So today I'm going to talk to you about algal blooms, and I realize, here's the first technical snack. Perfect. It's a good start. There we go. Yay, done. Yes. All right, about microbial plankton ecology and some of these things that you might not necessarily think very lovingly of. So when I was here last time, I saw this really beautiful talk about pollinators and spring and blooming. And typically when you are in terrestrial systems, the first thing that comes to mind, there's absolutely nothing wrong with spring blooms. There's nothing wrong with blossoms, with all the spring activity that's going on. Well, as soon as you transfer that to the ocean or the freshwaters and you think about blooms, people don't tend to think very positively about that.
Pine pollen, you know. There is pine pollen, so all the terrestrial blooms are...
All right, so allergy people, put that aside for a second. I have those two since I moved here, but aside of that, I think we can agree that the positive effects of that kind of like overweigh. I want to just put up this slide really quickly because I never want to forget about that. This is not me. This is many contributors to some of the different things that I'm going to mention. Included are people that are here, some engineers, faculty within the department. I work with Hans Perl's group at IMS, Chapel Hill. These are some people back from California, because I'm going to talk also about a marine toxic algae that has gotten a lot of press lately. And then obviously the core of my lab, which is Dan Vilti, who's not here, is getting ready for his committee meeting next week, and Gabrielle, who is here, and then our new tech, Marco Valera, is already pushing a lot of weight in regard to all the harmful algal bloom work. And then a lot of undergraduates that have in many ways contributed to this, including Alison Fowler, who have lost to the pollinator world. I really tried to keep her in marine science, but what can I do? Also on here is these tremendous group of agencies and institutions that have helped us do this. I want to point out Sea grants. I want to point NOAA funding. Sometimes these grants are very little, but they get you so far. And I can probably assume that many of you in here are very concerned with what is happening with some of those fundings. So I can only say most of what I'm going to present today in regard to North Carolina is heavily supported by NCC grant. And then also we work with a lot of the monitoring agencies here, NCDNR, and then there is some funding partially that fills in with having Gabrielle funded through the Climate Center, because she's our green thumb in the lab. She keeps all those cultures alive, and that's actually a pretty great skill. So are algae bad, right? They look cute, right? So in consistency with what we just heard, that they are the base of the food web. There's what the plants and grasses and trees are in terrestrial systems. No algae, no whales. No algae, no fish. No algae, nothing we can take out of the oceans. That is what is needed in these systems. Now let's think about how many there are. If you jump in any sort of freshwater or into seawater, you might want to keep in mind what's in 5 mils when you open your mouth. There's 100 million viruses, 10 million bacteria, and 5,000 of these algae or phytoplankton. Again, most of them are completely harmless, they're beneficial, they're the base of the food web, but a few are not, and they're getting a really bad rap. All right. If you can read, thank a teacher. If you can breathe, thank a phytoplankton. I don't think I have to drive that home too much. We all know that some of the, or a large proportion of the oxygen that we're able to breathe comes from those little critters. So the other thing that I want to drive home is they might be microscopic, but but they're beautiful. These critters are so diverse, their forms, their ecology is as diverse, and they can be really pretty to look at. Right, here's just a little plethora stolen from web pages. This is what's left of some of these algal cells when you stripe them off, and this is the skeletons that are left, and you can see at these forms, some of them are cellulose plates. These little guys are called coccolithophorids. They have these little calcium carbonate plates. They can also come in very different morphologies. And then here, diatoms, who actually make a little glass house or glass frustule. So unbelievable variety, beautiful to look at, very diverse. Also keep in mind, this is really high magnification. Typically these things are not more than 10 or 20 microns. And if you use the height of a dime and you slice it 1,000 times, you get 1 micron. So keep in mind they're small, but again, they're very abundant and really important. All right, so how do they do what they do? Well, they have the wonderful ability to reproduce asexually. What that means is as soon as there's a window of opportunity, again, think of what you try to keep your flowers growing in the yard. You give them nutrients, you give them light, and they sprout. Same for algae. There's nutrient input, there's enough light, typically when the spring starts, and they just bloom away. 2 cells make 4, make 8, make 16, and you get from a couple per liter to millions per liter within a few days. And when that happens, the pigment that you see inside of these cells starts to stain the water, and this is what you typically see when you see a red tide, a green tide, a mahogany tide. There's many names for these kinds of phenomena. How many of you have seen a tide in the ocean or in freshwater? I should probably ask how many of you have not, because most of you have either waded through one of these or see them on the beach, right? So again, these little critters, this is actually a little red tide species that was collected right off the California coast. This is not a culture. This is just straightforward water taken out and looked under the microscope. And you can see they're really thick and brown, and these guys are really happy. They're dividing, they're blooming. This is the end result. These are some of the pictures that you typically see in your textbooks. This little guy, so you see the magnitude of these blooms. These blooms are caused by those little coccolithophorids, those tiny guys with those beautiful armored plates. They are so tiny, but they can make these huge blooms that you can see from space. One of the reasons why you can see this milky substance or the milky bloom forming is because those calcium carbonate plates can be detected because it breaks the light, and the satellite imagery can pick up on that. Then something that people are typically more used to are these red tides. You can see that they are often in patches. Right here you see the normal ocean, but some of the currents, and these little guys are also motile. They can accumulate in really thick slicks. And then some of these algal blooms, cyanobacteria, that you typically see in fresh waters. So this is how these tiny organisms are able to color entire regions square kilometers in scale. They can also be fun. Sea foam, a lot of you might have seen this in little patches along the water. What happens when these millions of cells shred along the beach, the salt and the mixing, they produce these bubbles where all the cell contents and the polysaccharides start to mix up. If you ever bored, go online and Google and look for videos with sea foam where total coastal towns are completely overwhelmed and people are not opening their windows because everything is foam. Another thing that people might be familiar with is when these things bloom, specifically dinoflagellates, they can be extremely bioluminescent. Again, when those little cells share, they're triggered to release light. So these are some of the phenomena that we think more lovingly about when we think about algal blooms. So what's the harm? I think I made a pitch for algae being a really good thing and really cool. Well, the problem is globally, there's an increase in the frequency, the magnitude, and the geographic extent of harmful algal blooms. A little example here, what this map shows you in 1970, these red little dots show where a certain type of algal poisoning had been recorded, in this case paralytic shellfish poisoning, which is also a dinoflagellate. And here you see 2009, and look at these dots. Not only are we getting better in monitoring for them and detecting them, but they truly are increasing. What are the concerns? They're kind of obvious. We have all sorts of water quality issues. We have issues if there's toxins involved, because some of these harmful organisms actually do produce toxins that have an effect on us and animals. And then there's all sorts of things in regard to socioeconomic impacts. Who wants to drive their boat in the vacation on green slime? Nobody really wants to. As soon as these algal toxins or algal blooms show up, people are not sure if they can eat the seafood. So there's all sorts of halo effects that are harmful in areas where these blooms regularly occur. Some of the numbers are $80+ million per year that can be tied back to direct or indirect effects of algal blooms. So a lot of money involved. How can they be damaging? Here's a couple of more less harmful ways, but still potent. What you see here is a planktivorous fish, could be anchovy or sardine. They're planktivorous, that means they open their gills, they go through the water, and they filter out their food, the algae. What happens if your critter happens to have one of these silica or glass balls and sticky little ends on it, these spines like this one, they can actually get stuck in the gills, start to hurt the tissue, start to muck everything else up, and those little fish might actually suffocate from that. So there's some mechanical damage that has been reported. Another way that they can harm, this was in 2007, they found that there was a bloom of this little guy, we call it wisdom toothy, it's Gymnodinium cashiva sanguineum, sorry. This guy, if it blooms, it also makes the sea foam, but what they found out, that if seabirds swim through it, the chemicals associated with this foam, they completely adhere to the very inner layer of the feathers. They're surfactant, and they keep the birds from being able to keep that fluffy pillow of aeration that keeps them from getting cold. So they had flocks and dozens of migrating birds on the US West Coast that suddenly died of hypothermia, which was like, for other stresses, just one more thing that was added on. So some of these indirect effects are only now really coming up when people look more at these different events. A more obvious one is fish kills. So I told you that phytoplankton produce oxygen, but if you think about a harbor or an embayment, when they start to decay, the bloom is over, they run out of some nutrient, they start to decay, they start to get degraded by zooplankton and by bacteria, and they respire a lot. Also, obviously, algae respire during the night when there's no light for photosynthesis. Now, this can take all the oxygen out of the environment, especially if there's not a lot of current or exchange going on. And what that does is that some of the larger invertebrates, if you get to the 2 milligrams per liter threat mark, they suffocate. In this case, this was a fish kill in Redondo Harbor in California. They had more than 1 million ton sardines that they were shuffling out. What started to become really an issue is when they start to sink to the bottom, then they start to degrade, then they start to bloat up, and then they come back up. So these events can really harm a whole environment for several weeks until people shuffle them out. Some people in the marina actually started to skim them when they were kind of still fresh and threw them in the freezer. After we tested some of them, we told them you might not want to do that. There were also algal toxins involved. So besides maybe you don't want to necessarily put something on your Barbie that was already dead, there could be other implications. And then it was one of the LA harbors, so don't. There's other stuff. So that brings me to algal toxins. Here's a really easy cartoon. What happens? You have these guys bloom. Sometimes you can see that. Here's a water sample. You see in this case a very brown, dark color from the algae. Little critters like copepods and crustacean type, shrimp-like organisms filter them. We have our anchovy that just go through the water and accumulate these cells and within the toxins. And then one thing that is often a problem for us is shellfish. Shellfish are really good filterers. They have liters of water that they just pump through their bodies per hour, and that makes them really sufficient in getting the stuff out of the water column. Where does it go? Well, it goes up the food web. These are all different types of organisms that have been implicated with these algal toxins for trophic transfer. There's even whales that have showed significant amounts of some of these toxins. I'm not saying that the algal toxin might have brought the whale down, but it could have been that one additional stressor that really made a difference. And when I tell you how some of these toxins work on those that ingest them, that would make more sense. What we're concerned of is obviously when we're at the end of this food web. All right, why produce toxins? The two main biological functions that people typically talk about, grazer deterrence. If you're toxic, you don't get eaten. Well, that doesn't quite work out because we find it throughout the food web. Allelopathy, this is when one algae produces toxin, releases it to inhibit the growth of the other algae that might compete for the same resources or nutrients, right? People typically get those critters, put them in bowls, tweak them for certain settings, and sometimes it works out that it works that way, sometimes it doesn't. And then trying to extrapolate that to the field is really tricky. What are the triggers and trends? And this is where a little bit of the climate change comes in, and let me tell you right away, very little is understood about this at this point. We have natural river runoff and coastal upwelling. These are processes that bring nutrients into the water column. Blooms has always been there. This is a natural cycle, like I said in the beginning. However, what puts us in a really bad place is that eutrophication or nutrient pollution puts a whole other layer on top of what comes into the water column. Right here you see this nice little plume. It's probably really increased in nutrients and whatnot. If there is river runoff, all these nutrients make it into the harbor, and that might just trigger these algae that are just waiting for the opportunity. Climate change is being debated a lot, but there's varying things that people have hypothesized in regard to how that might affect harmful algal blooms, and I see a little bit in a second. Here comes what I would call my concern slide from a scientific perspective. Why is it hard to finally say what the real trigger is? These ephemeral events, they happen and they go. If I'm not out there all the time, I can't catch them. They're patchy. These things accumulate. They're not heterogeneously distributed, so it's, again, hard to capture them. Not all species are toxic. They might look toxic, but they're the other strain that can produce toxin at the same time, so that's hard to resolve. Even if they're potentially toxin producers, they don't produce it all the time. Some species produce several toxins at the same time. And then on top of that, if you think about climate change and long-term trends, there's a huge lack of long-term data to actually look at these tendencies in harmful algal bloom dynamics in comparison to long-term climatic trends. But it's complicated, but we're making a lot of progress. We've found a lot of things of these, and our tools are really getting better to look at these things. And I want to make two real quick cases, depending on how I make a time. Okay, all right. I'll make them very quick. In regard to some of these phenomena tied to a specific group of algae. What you see here is a map that indicates where some of these poisoning events have happened, and because typically the way they affect us is through shellfish, that is typically what gives them the name. So there's paralytic shellfish poisoning, amnesic, neurotoxic, ciguatera fish poisoning, and then we have our cyanobacterial blooms, which are freshwater, and obviously the Great Lakes and the brackish waters. The two examples that I want to talk about real quick is amnesic shellfish poisoning, and where I've studied this for a long time was along the California coast, and then we'll round up real quick with cyanobacterial blooms. There's not a lot of color here. Don't be fooled. People haven't looked a lot yet. We are one of those groups that are starting to look, and we're seeing some things, but we're not quite sure if toxin is involved with them. All right, Pseudonychia is this beautiful diatom that has these glass frustules. These chains, this is one of these cells that are nipped back to back. They form chains. They can be as long and thin as a hair, so when you look really carefully and there's a bloom against the light, you can see a little bit of that in the water. These guys produce domoic acid, which is known for amnesic shellfish poisoning. It can, depending on the dose you get through your seafood, it can cause nausea all the way to death. There were 3 recorded deaths in Prince Edward Island in Canada in the 80s. Since then, especially in the California coast, they monitor very vigorously in shellfish, and they close whole shellfish regions if they find certain amounts of this toxin. What it does is domoic acid looks very close to glutamic acid. It gets into your synapse, sits down, fires the synapses until the tissue disintegrates. It's irreparable. If you look at a sea lion that was hit by domoic acid and you look through his hypothalamus, you can see those huge lesions due to domoic acid poisoning. Where can you find it? Again, along the California coast. Oh, look, there's dots in our area. They're coming up, and pretty much when you look, you're probably going to find it. These were some of the usual suspects in California that we worked with marine rescue centers with. This was one of the Females, they came in, a 300-pound sea lion that had one of the highest levels of domoic acid in its serum, in its blood, but it completely recovered and was released. Typically, they come back in droves because the females tend to feed when the blooms are at the highest and most toxic. And at that point, they still have a couple more weeks to go with their fetuses. So often with the females, if they get domoic acid poisoning, they lose their pups because they have seizures, and typically that causes them to abort. A lot of seabirds, different species, were affected in California, and this is just showing, the gray area shows you all marine mammal strandings. The red area is those where we confirmed that they had domoic acid inside of them. And then the green is the peak of the algae and the toxicity in the water column. What this should tell you is sometimes we were able to detect the toxin in the water before the first sea lions started to strand. In other cases, they were calling us and telling us, I think a bloom is going on. This is how fast it goes through the food web. Now this is a little graphic, but this shows you a pelican affected by domoic acid poisoning. They can't stand up straight. They can't bring their head up. They start to tilt backwards. They don't keep any liquid, and this bird could not be rehabilitated. And you find similar symptoms in the sea lions that get hit by this. So it's a really potent toxin, and if it takes out a 400-pound sea lion, I think it should make us think, because they typically eat the same kind of seafood sources that we depend upon. Unfortunately, in LA, when people see sea lions on the beach, they think that's a good photo op. If they are behaving weirdly, you might not want to put your 2-year-old right next to it to take a picture. Fact or fiction? How many of you know Hitchcock, The Birds? Excellent. I see even young people raising their hand, which is awesome. This is actually based on a report in North Monterey Bay that they had about birds completely freaking out, crashing into things, normally behaving. They were able later on to tie this back to the diet of pseudonychia and domoic acid in those birds' diets. That was one of the first indirectly recorded incidences of domoic acid poisoning. So this has been around for a really long time, and now we're starting to figure out a little bit more about it. This was last year. This was one of the biggest yet recorded pseudonychia outbreaks. What you see here is Baja California all the way up to the Alaska coast. This greenish color indicates chlorophyll or algal biomass in the water. They went out and sampled that, and that was all mostly pseudonychia. What went through the press was that there was the biggest outbreak throughout the food webs. They found whales stranded, and in the end they had something like 138 million in disaster relief for seafood and shellfish growers in the area. That was the impact. One of the new things that they found is that there was a lot of domoic acid actually in benthic organisms. If you think about it, the algae are going to be in the water that is lit by photosynthesis. But what happens is when they degrade the algae, they start to get sticky, they clump, and they settle in the water column. And this can actually again make it to those different shellfish, Dungeness crab. People were very unhappy. They like the Dungeness crab. They couldn't eat that until mid-2016. So months later, they still found domoic acid at threshold levels that did not allow for the seafood to be consumed. We did a little experiment, and we actually grew these toxic algae in our balls, and this should look familiar to Allison. This is what shows you what happens. First they happen in green, they start to accumulate, and then they make these particulates that start to sink through the food web and are a vector that goes directly to the benthic and the sediment living organisms, and they're very highly toxic. How am I doing? 5 minutes? Excellent. All right. Quick switch to cyanobacterial blooms, which you probably have heard a little bit more recently in the news about. Now these are prokaryotes, but they're also considered phytoplankton because they're primary producers. And most of you probably have heard about the Toledo water crisis and Lake Erie in 2014, where they had to shuffle in 10,000 of gallons of water because the drinking water suddenly became completely unsafe due to cyanotoxins within the system. This was Lake Okeechobee in June 2016. Florida is battling this issue really strongly at the moment, and there's all sorts of reports. You just need to Google this and you will find all sorts of reports on this. However, you don't have to go far. This is Joan County. This is near Edenton Bay. This is the Albemarle Sound. What are the toxin producers? Here you see them. They're really morphologically diverse. You have these colony-forming, glob-forming organisms, and then these filamentous cyanobacteria. Here's a short list of the toxins we know that are there. They can be hepatotoxins, cytotoxins, endotoxins, dermatotoxins. They can be neurotoxins. All 4 of these groups have been found in some small amounts in some of the water bodies around here, and we're one of the groups that is looking into that. That doesn't need to be alarming because they have to have a certain level that truly has an effect on humans. The thing that is tricky here is microcystin is not just microcystin. There's 100 different congeners of it. Also, so for example, microcystin doesn't just produce microcystin, but it can also produce 2 or 3 of the other toxins. So it becomes really hard to tie down on what conditions specifically trigger which organism to produce which toxin. It's a challenge. These are some of the reservoirs and sampling sites that my new tech becomes to get familiarized really much, where we try to do some NCC grant work and work with DWR. We are working in the Cape Fear River with Hans Perl's group, where these water treatment plants are using some of our in situ toxin elements. These are called spat bags. They look like little resin bags or tea bags. This is when you put them in. This is when you get them out. What they do is they accumulate dissolved toxin, and you can then wash that off the resin and get an accumulated signal. This is good for 2 reasons. First off, it gives you low thresholds, which you don't get if you just go out, take one sample from one time point. They typically miss that there's any toxin in there. This is an accumulated signal over time. And the second thing that is good for it, you can use it to look at all 5 major toxins that are currently being discussed. We do it in High Rock Lake, we do it in Jordan Lake, so there's several different water treatment plants and reservoirs, and now also the Jordan River, where we're screening for these different compounds. Future trends, global climate change. I know I'm almost done, and now I'm coming to climate change. I know that's exactly what Walt is thinking. Here's one of the major issues. Most of the general predictions people make are based on one case, meaning one algae in one specific region, and you can't not use that to extrapolate generally. You can't. These critters are as different as you are from your neighbor. They're not the same thing. They might fall into the same phyla, but they're very different. So it's complex, and then there is not just temperature that is increasing, there's not just ocean acidification in the marine systems, there's not just pollution. All of these factors have antagonistic, synergistic effects on different critters in different ways. So it's a little harder to decipher that. And then there's also many trophic levels that need to be considered if we're thinking about how we would get exposed to these toxins. Are we eating seafood? Are we drinking the water? Are we swimming through it? So it's not just one answer. Not going to go into this in detail because this is what I just really summarized in regard to how hard it is. But where I'm really excited is that we're finding these interdisciplinary approaches. We're trying to go from the molecular single cell and its toxin content to something like remote sensing to put it all together. We have better models now because we're starting to collect these long-term data sets that actually allow us to look at climatic trends. And this is a huge one. Public awareness and outreach. Most of our sampling would not be possible without the people in the Jordan River who go out on their pier, put a spat bag on, take one off, and throw it in the freezer. We have mainly public citizens and people that are in those task forces that help us do this, and they're concerned because they have their kid running on the pier and jumping into the green slime. They want to know why their kid coughs, and they can help us get that data, which we could never afford to collect if we were by ourselves. I had to put this in. This is really alarming. We're just starting to do this, and now everything that comes about clean water, safe water issues is taken almost away underneath our feet. This is really scary. This is, I think, at this point, the biggest obstacle. I can deal with all the complexity and the food webs and pulling together different interdisciplinary sides to the problem. This, nobody can do about this, and this is so important, especially because these funding agencies make this link to public and outreach possible for us. Well, so we should care about all of these issues. This is a red tide phenomenon with manatees on the Florida coast. Red tide has shown up here once before in the 80s. So climate change, increasing water temperature, expansion range of red tide, that would be really bad for the Florida tide type. Pelicans, this. And then in the middle, as far as freshwater is concerned, if you have a pup, don't let him go into this water and slurp a sip. Don't. It's not good. Don't do it either yourself. All right. And this is thank you.